Hello everyone, welcome to today's live broadcast, Advances in AMPK and Autophagy Signaling. I am Brenda Kelly Kim of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals, and sponsored by Cell Signaling Technology. Cell Signaling Technology is a private, family-owned company headquartered in Danvers, Massachusetts, with over 400 employees worldwide. Active in the field of applied systems biology research, particularly as it relates to cancer, CST understands the importance of using antibodies with high levels of specificity and lot-to-lot consistency. It's why we produce all of our antibodies in-house and perform painstaking validations for multiple applications. And the same CST scientists who produce our antibodies also provide technical support for customers, helping them design experiments, troubleshoot, and achieve reliable results. See more at www.cellsignal.com. Before we start, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. You want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear this presentation properly or see it, let us know by clicking on the support button on the top right or use the Q&A button, and we'll try to make sure to resolve any issues. Please note, this webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Now let's get right to today's presenter. We are proud to welcome Dr. Reuben Shaw. Dr. Reuben Shaw is a professor in the Molecular and Cellular Biology Laboratory at the Salk Institute. Dr. Shaw's lab focuses on the AMPK signaling pathway, which coordinates metabolism and growth at the cellular and organismal levels across eukaryotes. This pathway is inactivated in a number of human cancers and altered in metabolic disease. The Shaw lab has been using a combination of biochemistry, cell biology, and genetically engineered mouse models of cancer to dissect the role of the LKB1 AMPK tumor suppressor pathway in coordinating metabolism, autophagy, and cell growth. This work has led to the identification of a number of new direct substrates of AMPK, which provide a molecular basis for how cells reprogram their metabolism and growth under conditions of nutrient deficiency. The Shaw lab also utilizes genetically engineered mouse models to explore how reprogramming of glucose and lipid metabolism contributes to tumor genesis and to develop and test novel therapeutic approaches for multiple types of cancer. I'll now turn it over to Dr. Reuben Shaw for his presentation.
Great. All right, welcome everybody. So I'm going to talk to all of you today about the center focus of my laboratory and a metabolic pathway that actually links the both acute and long-term effects of metabolism to growth control, and therefore links different diseases ranging from cancer to diabetes. So the central focus of my lab centers around those two very basic cellular processes. How is cell metabolism and cell growth coordinated at the molecular level? And out of our studies, as we decode new components of these signal transduction pathways that modulate these, we're always looking for new avenues and new targets to treat both cancer and metabolic disease. Because as you're about to hear, in this case, this particular signaling pathway is altered in both of these very distinct clinical diseases. So the focus point of my lab's work and the focus point of much of today's talk starts with this very basic signaling diagram that you can see on this slide here. And this involves a highly conserved heterotrimeric protein kinase complex known as the AMP activated protein kinase which you see here in the center of your slide. Across all eukaryotic species, as you can see at the bottom of the slide, so from budding yeast all the way back through plants, flies, worms, and mammals, there is an ortholog of AMPK and its associated subunits. And in all of those species, they dictate different features of metabolism. So both glucose and lipid metabolism, how cells uptake glucose and lipid and utilize it and store from single-celled organisms all the way through specialized metabolic tissues in mammals, all of them engage this same pathway. So it was some years ago now when I was a postdoctoral fellow in Luke Cantley's lab at Harvard Medical School, working on not a metabolism gene, but in fact working on a kinase altered in cancer, that I made the unexpected finding that kinase, LKB1, was in fact the long sought after upstream kinase that directly phosphorylates AMPK and is required for its activation. Now, what was well appreciated in the field was that this critical phosphorylation event is stimulated under conditions when cellular ATP levels are low. So in mammals, this occurs under conditions of low nutrients, including hypoxia, so low oxygen or low glucose. It also occurs in response to a wide variety of mitochondrial insults. So anything that inhibits OXPHOS, electron transport chain inhibitors, including classic drugs such as antimyosin, rotenone, CCP, anything, you don't even have to have full uncoupling of the mitochondria. Even mild insults to the electron transport chain will decrease ATP output and therefore decrease intracellular ATP levels, resulting in the activation of this kinase cascade. So under those conditions, LKB1 directly phosphorylates and turns on AMPK. However, in higher species such as mammals, this is not only activated in the cell autonomous way by nutrients or mitochondrial insults, but hormones induced in response to exercise as well as caloric restriction also serve to activate this 2-kinase cascade. One of the reasons there's been great interest in AMPK over the course of the past 15 years actually came from studies first demonstrating that the widely used type 2 diabetes drug metformin in fact acts as a complex 1 inhibitor. Complex 1 of the OXPHOS chain is actually inhibited by metformin and related compounds. This decreases ATP output, and while the details and the relative importance of different downstream effectors are still under very active investigation by many labs, what is clear is that in all systems, metformin and related compounds robustly activate AMPK, and that plays at least a partial role in mediating the benefits of lowered glucose and lowered lipid in response to metformin. Besides metformin and related compounds, there are other ways to activate this circuit pharmacologically. There is a cell-permeable AMP mimetic known as ICAR, which you can see on your left-hand side. Again, that will directly bind to this AMPK heterotrimeric kinase complex, favoring its phosphorylation then by LKB1. If you inhibit glycolysis in tumors or in cell types that really rely on glycolysis for their ATP production, you'll also activate this circuit. And then finally, over the course of the past decade, a number of different pharmaceutical companies, first starting with Abbott Laboratories and their compound A769662, there are a number of different derivatives of this compound and other naturally even occurring compounds, such as salicylate, that directly bind to actually an interface of the beta isoform of AMPK and activate this kinase complex independently of intracellular ATP levels. So there are a number of different tools then that the field uses to explore the relative benefits and downstream effects of activation of AMPK itself. This circuit, as I said, gets activated under conditions when intracellular ATP levels are low. And you can really think about this as a checkpoint. So many of you may be familiar with the DNA damage checkpoint that is activated when cells receive damage to their DNA. Checkpoint kinases in that case get activated, and then they phosphorylate a number of downstream targets to help repair the DNA and stall the cell cycle while they do so. In this case, the insult to the cell is loss of intracellular ATP, and so under those conditions, AMPK is activated in an LKB1 dependent manner, and AMPK wants to immediately, rapidly shut off ATP-consuming anabolic processes. So all the major biosynthetic processes, protein synthesis, lipid synthesis, get shut off. But it's not enough to stop the bleeding of ATP from the cell. You need to actually restore ATP levels back to normal. And some of the ways that AMPK does so is through the stimulation of fatty acid oxidation, and as you'll hear a lot about in today's talk, through the stimulation of a cellular process known as autophagy. Now, genetic and pharmacological studies from a number of labs, including my own lab and our collaborators and the studies that are highlighted in blue here, have gone on to study the role of AMPK in different specialized metabolic tissues in mammals, so in both knockout genetic manipulation studies in mice or pharmacologic studies in mice and in lower species. We and others have shown that activation of AMPK improves blood glucose and improves lipid content in the liver. And conversely, if you genetically remove AMPK or its upstream kinase, you get hyperglycemia if you remove the upstream kinase, and hyperlipidemia has been observed by other laboratories. Conversely, if you gain AMPK activity pharmacologically, you can stimulate exercise, and this is shown in a collaboration between my lab and Ron Evans' lab. You can extend the lifespan of the roundworm C. elegans, and that was work between my lab and Andy Dillon's lab here at the Salk. And AMPK has been hypothesized and shown in a number of studies, a limited number, but some very nice studies, to extend the longevity in flies and to mediate different features of caloric restriction, and actually in a wide variety of species. So that's another very active area of research, which is really the subject of a whole different intense talk. Finally, because in mammals, much of our nutrient sensing capacity actually relies within different neuron populations of our brain, others have studied the role of AMPK in glucose sensing neurons and in other neuronal functions where it also plays critical roles. Finally, if any of you are listening and hoping to find out more about new super enhancing drugs, it turns out that people abusing AMPK activators are commonly found amongst professional athletes, as improving your metabolism, of course, is one way to improve athletic features. And in fact, at both the Tour de France and at other sporting events in Europe, various coaches and players have been arrested and tested positive for literally carrying some of these compounds that I alluded to, which are bought from standard chemical houses. So it's quite remarkable that not only are researchers in this field working on new therapeutics for diabetes and cancer, they're also accidentally discovering the latest new drugs for doping. Finally, given the initial connection from my and other work of cancer to this central metabolic enzyme, this raises the question of whether or not other drugs, such as metformin or ICAR, when taken by diabetes patients or by other patients, whether or not this suppresses one's risk of cancer, and that is another very active area of research, which I won't have time to talk about today, but if you're interested, I encourage you to check that out. There are more and more better detailed epidemiology and now clinical trial studies of metformin in different subtypes of cancer. And the long story short is that it appears, as is true for nearly all types of cancer and their treatment, that certain subtypes maybe gain benefit, and many types of cancer will see no benefit at all. Okay, so given that seemingly almost incredible list of benefits that are gained from the activation of this protein kinase, there has also been interest in figuring out which, first of all, how on earth does one enzyme mediate these very disparate seeming phenotypes, from fighting aging, suppressing cancer, suppressing diabetes. Is this all through a handful of specific pathways, or are there different subsets of downstream targets that mediate different effects? And this is, in fact, the major focus of my laboratory. So using some of those genetic models that I alluded to in mice, we have set out to dissect what the key direct substrates, the proteins directly phosphorylated by AMPK in different cells and different tissues are. And so one reason to do that is not simply to identify new therapeutic targets, but beyond that, even developing a better tool set to be able to track the activity of this pathway in response to existing therapies and in response to different disease states is really critical for developing a more detailed understanding of how cells adapt their metabolism and adapt their growth in response to different physiological and pathological disease changes. Okay, so one of the central techniques and central pursuits of my lab has really been using a combination of phospho-proteomics and bioinformatics to identify new direct substrates of AMPK. And without getting into the fine details, we used a peptide library positional scanning methodology developed by a professor named Ben Turk at Yale University when he was also a postdoc and was Cantley's lab at Harvard Medical School. This method allows you to get to use degenerate peptide libraries to identify the optimal sequence that a given kinase likes to phosphorylate. In this case, AMPK is a very selective kinase and requires 4 or 5 different specific residues relative to a central phosphoserine site that is targeted in order for something to serve as an AMPK substrate in vivo. As you might imagine, every time you start restricting and imposing that it has to be particular amino acids relative to the serine, that means the number of proteins in the database that contain such a sequence is more and more restricted. However, I should point out that if you look within the human genome and ask how many proteins contain this so-called optimal AMPK motif, the answer is around 2,000 proteins contain this sequence. If you then started cloning them one by one and asked how many of them actually serve as AMPK substrates in vivo, in cells, or in tissues in mice, the answer is less than 1/10. So the presence of the sequence appears to be required, but it by itself does not, it's not sufficient to tell you what is an AMPK substrate and what is not an AMPK substrate. So in the first part of the screening method, we used bioinformatics to kind of generate that list of 2,000 prospects. But the real area where we hit pay dirt was using biochemical fractionation, taking wild type and genetically knocked out AMPK deficient cells or AMPK deficient tissues, or LKB1 wild type or deficient cells or deficient tissues. We also have the extra benefit that we know that this pathway is activated only in response to metabolic stress. So as shown in this example, in actual mouse livers, we can treat mice with the diabetes drug metformin, which induces ATP loss within the liver. And then we can use an affinity tool such as this phospho-motif antibody, such as cell signaling cells, in fact, for the AMPK substrate sequence shown up top here. And you can actually immunoprecipitate proteins out of a total cell lysate that bind to this antibody. And so you can see only in response to an energetic stress, you get particular polypeptides shown here by the red arrows, and you only see those in wild-type livers treated with metformin and not in the knockout livers treated with metformin. Another tool that we've used is taking advantage of a natural phosphopeptide binding protein known as the 1433 family of regulators. 1433 proteins only bind to phosphoserines that actually also have an overlapping consensus with AMPK. You need to have a basophilic, you have to have an arginine or a lysine in the -3 residue. And so that overlap between what 1433 likes and what AMPK likes means that we can actually pass lysates over a column of GST as compared to GST 1433 and ask out of a wild-type liver, for example, what binds to 1433 as compared to what binds to 1433 out of an AMPK knockout liver. And much of that work is not yet published, and so I won't be talking about that today, but we've done that even using stable isotopic labeling in mammals with John Yates' lab at the Scripps Research Institute up the street. But to summarize really the first 10 years of work out of the laboratory, we've decoded a handful of new direct substrates of AMPK that we believe are critical for AMPK's ability to regulate different aspects of cell growth and cell metabolism. So amongst them, we've identified phosphorylation sites in the tuberous sclerosis 2, TSC2 tumor suppressor gene, as well as the protein raptor, which is a direct binding partner of the mammalian target of rapamycin. And these two events couple AMPK activation to the inhibition of mTOR activity. So this is now very well established from many labs and actually from many species. Across all of those eukaryotic species, starting with budding yeast, an AMPK ortholog inhibits mTOR function across all of those species. And there was actually a very nice paper in eLife just recently demonstrating in budding yeast that the raptor homolog is in fact directly targeted by the AMPK homolog SNF1. So I would encourage you to check that paper out if you're interested in that evolutionary conserved link. In a sense, this is one of the most basic ways that all eukaryotic cells know whether they have food or not. If you have food, then mTOR activity is on and the cells are allowed to grow. If you are low on nutrients, a version, an ortholog of AMPK gets activated. It directly phosphorylates a raptor homolog, a core component of mTOR, and immediately shuts off the central engine of all cell growth. We also have gone on to show that other direct effectors of mTOR, including the SRBP1 transcription factor, are phosphorylated by AMPK on distinct sites. And one of our more recent discoveries in that area, which will now become the focus of the rest of the talk, is actually in a kinase that in budding yeast is known as ATG1, autophagy 1, and in higher eukaryotes is known as ULK1. So autophagy is a highly conserved cell biological process that occurs basally in many different cell types within the body, and it is a mechanism for the cell to clear both protein aggregates as well as dysfunctional organelles. So outside of the classic proteasome that targets many proteins for degradation, this is the other major cellular disposal destruction mechanism, which is this classic autophagy pathway. The proteins involved in autophagy were originally worked out in genetic screens in budding yeast, and they were all given the suffix ATG. So as I was just telling you, the very first gene and the most upstream gene in the complex, interestingly, was called ATG1. And you can see in this picture that I have at the top of the slide, this is taken from mammals, so there it's called ULK1. And what's more notable is actually that this ULK1 is actually the only core component of the autophagy pathway that is a serine threonine kinase. So all of the other components of the canonical autophagy pathway include different types of scaffolds or lipid conjugating enzymes, or variants, in fact, of the classic ubiquitination cascade in E2s and E3s. But lying at the head of the pathway, the gatekeeper decision, in fact, funnels through this serine threonine kinase known as ATG1 or ULK1. And so it was therefore of great interest in my lab many years ago now when one of the first graduate students, Daniel Egan, unbiasedly identified ULK1 as one of the most highly regulated 1433 binding proteins induced in response to AMPK. And we believe that it was one of the most differential hits because, in fact, AMPK phosphorylates at least 4 different residues within ULK1. ULK1 has a kinase domain at its N-terminus, and it has a conserved C-terminus by which it scaffolds to other regulatory subunits. In the middle is this kind of disordered linker region, and that is where AMPK induces the phosphorylation of ULK1 on highly conserved AMPK phosphorylation sites. As I alluded to, we map these sites, and all this data that I'm showing on this point is actually published in the supplement of that science paper that was on the previous slide in the bottom left-hand corner. So I encourage you to look it up if I'm talking too fast and you are interested in these details. So contained within the data of that paper are the 4 different AMPK phosphorylation sites that we identified, 2 of which are well conserved in a ULK1 homologue known as ULK2, and both conserved at least back through upper eukaryotes. One of them has a perfect both AMPK phosphorylation site and a perfect 14-3-3 binding site, and that is this serine 555, to which we generate a phospho-specific antibody in collaboration with cell signaling technologies, and this antibody is now commercially available and widely used by the field. So this is a great readout of whether AMPK is talking to the autophagy complex. So back at the time of that study, we did some old-fashioned classic biochemistry and proved that recombinant AMPK will directly phosphorylate ULK1 in a test tube using classic P32 in vitro kinase assays. For example, you could compare the stoichiometry of that phosphorylation to how well AMPK phosphorylated Raptor, a well-studied substrate. Raptor has 2 AMPK phosphorylation sites, and you can see if you actually look at the protein abundance here, judged by the MIC tag in the left-hand bottom, that actually ULK1 is much more highly phosphorylated than Raptor is in vitro. As I said, in collaboration with cell signaling, we raised antibodies against these sites in ULK1. There's that serine 555 antibody. There's also a serine 461 antibody, and you can see the recombinant AMPK in vitro induces ULK1 phosphorylation on those sites. More importantly, we proved that we can use such antibodies to read out the activation of ULK1 by AMPK endogenously in different cell lysates. So on the upper left-hand side here, you can see looking in hepatocytes that are wild type or ULK1 knockout, we can see the induction of endogenous ULK1 phosphorylation here in response to metformin. So the widely used diabetes drug metformin is actually inducing the phosphorylation of ULK1 within an hour of treatment. And one prediction or question that raises then is for the 25 million type 2 diabetics that take metformin every day, whether anyone realizes it or not, they are, at the time they take the metformin pill, activating AMPK and activating ULK1 to initiate autophagy. So you're effectively activating autophagy in a synchronized fashion in response to metformin through this action. Now, how much autophagy actually gets induced and the relative importance of that autophagy is not known, and that's something that my lab is exploring right now, but it's still a very interesting thing to think about the pharmacology of this, and that you could be synchronously activating autophagy, and that may have benefits on the liver and on various physiological functions. So again, on the right-hand side of that same upper left panel, taking AMPK wild type or knockout hepatocytes, again we see robust phosphorylation of ULK1 on that serine site in response to metformin in hepatocytes. We see the same thing if we look in murine embryonic fibroblasts, so in MEFs, again ULK1 or AMPK wild type or knockout when you see robust phosphorylation of endogenous ULK1. Now, one of the biological phenotypes of being deficient in autophagy or deficient in AMPK then is actually the accumulation of distended and defective mitochondria, which does get worse over time, as you might expect. So when animals are first born, the number of defective mitochondria is minimal, but as one just lives their lives, you accumulate damage from a wide variety of sources, and that, in fact, leads to the accumulation of defective mitochondria, which you can see if you examine the paper closely. In a final point that's contained in that old paper, we showed that actually you require those AMPK phosphorylation sites for the function. So ULK1 deficient cells that have been stably reconstituted with a non-phosphorylatable allele of ULK1 as compared to wild-type ULK1 or a kinase inactive version of ULK1. The wild-type ULK1 restores proper mitochondria to these cells, whereas the kinase dead, the catalytically dead ULK1, or even the catalytically active version, but a version that cannot be signaled to by AMPK, that actually is completely defective for proper mitochondrial homeostasis. And we looked at this using other assays. Here we used MitoTracker to actually visualize the mitochondria. You can use a dye known as JC1 to look at mitochondrial membrane activity. And again, we see that you require ULK1 and you require the AMPK sites in ULK1 for proper mitochondrial function. A long-term biological consequence of that is that cells that are genetically deprived of core autophagy proteins, either ULK1 and ULK2, or a canonical component of the pathway further down called ATG5, if you put them under starvation conditions, it's been reported by many labs, including Craig Thompson's lab is one of the first large papers on this topic, that actually cells that cannot undergo autophagy, cannot cannibalize their own internal stores for nutrients, more rapidly undergo cell death. So autophagy is generally a cell survival signal that allows cells to live under nutrient-limited conditions. And if you genetically remove autophagy through removal of ATG5 or removal of ULK1 or ULK1 and ULK2, you simply increase apoptosis, as read out here through annexin 5 staining in a FACS assay, a fluorescent activated cell sorting type of assay. Importantly, we went on to reconstitute ULK1 deficient cells with, again, a wild type or kinase dead or non-phosphorylatable allele of ULK1, and we see very comparable effects to full deficiency. In other words, wild type ULK1 restores normal cell survival onto the ULK1 deficient cells, but the kinase dead or the non-phosphorylatable ULK1 are equally defective and cannot rescue cell survival in response to starvation. So the conclusion of all of that is that mutation of the AMPK sites in ULK1 mimics complete deficiency for ULK1. So you need ULK1 at least for starvation to induce AMPK, to induce cell survival. So to give some big picture context to this, then actually within the AMPK field it was already known that AMPK in lower nutrient conditions can actually trigger the formation of new mitochondria, so mitochondrial biogenesis, which is controlled transcriptionally. That was known to be under the control of AMPK already. So with this study we showed that the turnover of defective mitochondria is under the control of AMPK through its stimulation of ULK1. So we thought of this as kind of a mitochondrial cash for clunkers program where the cell trades in its defective cars, its defective mitochondria, for shiny new mitochondria through the same upstream signal from AMPK. Another very interesting and important point about ULK1 activity, though, is not only is ULK1 directly phosphorylated and activated on those sites by AMPK, but on different sites within ULK1, it is directly phosphorylated and inhibited by mTOR. So ULK1 then sits as a downstream node that integrates signals from both AMPK and mTOR. And this fits well with decades of studies of conditions that activate autophagy. As you can imagine, when nutrients are low, AMPK gets activated. It both directly stimulates autophagy, but by AMPK phosphorylating TOR, it also relieves this brake that also gatekeeps ULK1 activity. So it's really in a two-pronged mechanism that AMPK is stimulating ULK1 function, both by directly phosphorylating it and also by decreasing the amount that ULK1 is phosphorylated by mTOR. So critical questions in the field back at the time that we finished up that work were whether or not ULK1 itself would turn out to be a critical inhibitor of tumor genesis or inhibitor of diabetes in the same way that AMPK is thought to be. But then more directly, what is ULK1 phosphorylating to cause its effects? And if we could make a drug that inhibited ULK1 function, might that have utility within cancer? So as I told you, autophagy is a cell survival mechanism in general in response to stress, and in particular in response to nutrient stress. This was originally defined, actually in original papers from the Oshumi lab. Here with ATG1 deficiency, you can see if you starve them of nitrogen, the cells lacking ATG1 function completely crash within days. Wild-type cells deal with this stress in part by inducing autophagy. And I just showed you our own data from a number of years ago now that actually doing RNAi against ULK1 and ULK2, you get the same effect. Now, tumors, it turns out, also are metabolically restricted for nutrients, including oxygen as well as glucose, and they rely on autophagy for cell survival. And so now genetic studies, mostly from Eileen White's lab, as well as from Alec Kimmelman's lab, have genetically targeted the autophagy process. I should also add Kevin Ryan's lab in Scotland has done very nice work in this area, and shown that if you genetically remove autophagy, you get defects in different tumor formations. In this summary taken from a review article from Eileen White's group, and so I'll just direct you, if you're interested, to check out more about that. Her lab, in a series of papers over the past 5 years, has shown that combining genetic deficiency for the central autophagy gene ATG7, if you combine that in KRAS or BRAF mutant tumors, either in the lung or in melanomas in different settings, that you actually convert aggressive carcinomas into more benign oncocytomas is the clinical pathological term for these more benign tumors, which also have as a cardinal feature defective mitochondrial function. So this suggests that perhaps inhibiting autophagy function would be beneficial for the treatment of cancer. And in particular, the Kimmelman lab out of Harvard Medical School has shown that this is particularly at play in the context of pancreatic cancer. And again, I'll just suggest you check out papers from that lab. Beyond that, though, this same circuitry of mTOR regulating ULK1 will get engaged in response to a wide variety of anti-cancer agents. So not only mTOR inhibitors themselves, but also inhibitors of upstream inputs to mTOR, including AKT, PI3 kinase, or EGF receptor inhibitors. All of them will result in a suppression of mTOR catalytic activity. Because mTOR inhibits ULK1 activity, when you inhibit mTOR, or you inhibit any of these upstream inputs, you will dramatically and rapidly increase ULK1 activity and autophagy. That implies that all cancers that are being treated with targeted therapeutics of the kind that I list on the right-hand side will have at least an immediate response of the induction of autophagy via ULK1. That is a testable hypothesis, which has not been fully tested, in fact, yet, that comes out of this simple biochemical circuitry. So with that in mind, my lab, in collaboration with others, was interested in whether or not, if one developed a competitive inhibitor of ULK1, you would then lose the survival benefit that tumor cells gain when you treat with these cells. It's known that many of these drugs are in fact cytostatic, and therefore if you can convert their responses to a more cytotoxic response, that would be a great benefit for cancer therapy. Now I should add that autophagy is a very common mechanism. There are many studies now. This is taken from a review article from Cell Death and Differentiation from last year, listed at the bottom. So I would urge you to check that out if you're interested. But autophagy is a common mechanism now documented in response to many different anti-cancer agents. As I just suggested on the previous slide, they are now, have now been documented. They and many other styles of cancer therapies have been documented to induce autophagy. In response to that fact, many have been keen on drugging autophagy for cancer therapy, and as such, the only pre-existing compounds that inhibit autophagy are lysosomotropic agents such as the chloroquine family of compounds. So these are lysosome inhibitors, and therefore you're not selectively inhibiting autophagy, you're actually inhibiting all lysosomal function. Incredibly, that's actually tolerated in cells and in the body, and you can actually have therapeutic benefits from these agents, and that is, as you can tell, these are clinical trials in different types of cancer where chloroquine and chloroquine derivatives are being engaged. And again, here's the reference to this review article that talks about all these different clinical trials where chloroquine drugs are being tried. Now, to come back to where I was many slides ago, ULK1, as I said, is the only serine threonine kinase, though, so that means it is one of the only readily druggable targets within this complex. There are other proteins, particularly within the ubiquitin conjugating system, that are also, in theory, druggable, and there is great interest in all of these, as well as this lipid kinase VPS34 in the middle, which is also, in theory, druggable. However, VPS34 also plays central roles in vesicular trafficking outside of its roles in autophagy. So it's really important to try to find targets that are dedicated for autophagy, and also for the cancer context, you would only be treating patients for a short amount of time, and therefore you would not have the side effects and negative consequences of prolonged inhibition of autophagy. Okay, so as I told you, ULK1 helps promote cell survival by keeping cells alive under stress. So how do we go about developing a therapeutic? Well, we use those same peptide library screening methods that I told you about for AMPK, and I won't go through them now. This was recently published. We did the same thing for ULK1. Turns out ULK1 is also a very selective kinase, not quite as selective as AMPK, but interestingly and oddly requires hydrophobic residues in the -3 position. You can see methionine is highly selected for in these peptide libraries. And to make a very long story short, we recently published the results of this screening methods and identified a number of different ULK1 in vivo phosphorylation sites in other core autophagy proteins. We have not explored the functional effects of all of these different phosphorylation events, and in fact we focused on this one particular serine within VPS34, which is quite well conserved, and interestingly it appears to be the only ULK1 phosphorylation site in VPS34. What's also notable is that Kunling Wan's lab, also working on ULK1, identified a phosphorylation site in the VPS34 binding partner, Beclin, serine 15. We also independently identified that site, and what's most striking is that the sequence of Beclin that gets phosphorylated by ULK1 in their study also aligns well with the sequence flanking VPS34. So we think there is a ULK1 motif, so to speak, at least a loose motif, and that is in common between these two substrates. Interestingly, those two targets then are the very first to have commercial phospho-specific antibodies generated against them. A number of companies have made antibodies against the Beclin site, cell signaling cells, a nice antibody against this Beclin serine 15 phosphorylation site. They also sell an antibody that we developed in collaboration with them against the serine 249 site in VPS34. I'm going to keep talking about that site for the remainder of the talk, but so if you are interested in blotting for ULK1 activity in your system, these two antibodies, phosphoserine 249 in VPS34 and serine 15 in Beclin, are some of the more advanced reagents that exist. Collaborating with Nick Cosford's lab, Nick is a professor at the Sanford Burnham Previs Medical Discovery Institute down the road here in La Jolla, California. Nick's lab are gurus in medicinal chemistry, and so taking ATP competitive inhibitors, we serially examined and identified tool compounds that directly inhibit ULK1. And I won't belabor this since I'm short on time and this is all published now, but we screened through hundreds of different compounds to identify a lead tool compound which we published in this paper earlier this summer. This compound, SBI 0206-965, which I will refer to for the rest of the talk simply as 6965, shows quite incredible selectivity for ULK1. And that was one of the reasons that we focused on this particular compound. We had other compounds that also very nicely inhibit ULK1. They inhibit a number of other kinases. This compound was very selective towards ULK1, and I would just urge you to explore the paper for more of the data, such as this screening method from DiscoverRx that we use to actually examine the true specificity. So indeed, we profiled all 512 kinases in terms of their selective binding, and ULK1 indeed is a very selective inhibitor. This is a very selective inhibitor for ULK1. The selectivity, in fact, is so great that if you compare it to other ATP-competitive kinase inhibitors in the clinic, 6965 itself, even as a tool compound, is very favorable and has a profile very similar to different EGF receptor and BCR-ABL ATP-competitive inhibitors which are in the clinic right now. And it's even better than other ATP-competitive inhibitors which are also widely used in the clinic. So it's the type of compound one could use, at least in theory. We went on to use RNAi to prove that some of the other kinases, such as FAC, which is one of the other major kinases that it independently inhibits, that FAC is not regulating autophagy in any of the systems that we're looking at. Again, we believe that ULK1 is a key target for some of the effects of this compound. Here we are just showing that the compound, when treated in cells that are overexpressing ULK1, that you can actually suppress that phosphorylation event I told you about a few slides ago, the site in VPS34 or the site in Becklin. The phosphorylation of those is diminished when you treat cells with this 6965 compound. Another thing that's known about autophagy is that not only will starvation induce autophagy, but actually chemical inhibition of mTOR itself will induce autophagy. And here we're using this autophagy cyto-ID kit to read out autophagy within this particular cell type, A549 cells. If you treat with a chemical inhibitor of mTOR, such as AZD8055, you see the induction of these autophagosomes, which are in fact blocked by 6965 treatment. Finally, is another readout of ULK1 activity in the cell. There is a phosphorylation site in the ULK1 complex component ATG13, which can be read out here endogenously. It is increased in response to mTOR catalytic inhibition, and it is suppressed in response to our 6965 compound. So this is the third phospho-specific antibody that I've mentioned today that serves as a readout of ULK1 activity. I won't belabor some of this because we're running short on time, and I want to make sure that we have time to get to questions. But within this study, we went on to show that treating cells with this ULK1 inhibitor induces apoptosis only in the context of starvation. So if you treat most normal cells with this compound, they completely tolerate it. Inhibiting autophagy is fine if you have plenty of nutrients around, but if you put them into starvation conditions, then you induce very potent apoptosis, as you can see here. And you can see with other readouts on the right-hand side. However, tumor cells which as I mentioned earlier, have higher rates of basal autophagy than normal cells. And indeed, in tumor cells, as you can see in this dose-response curve, just escalating doses of 6965 by itself is sufficient to induce cell death, as read out by a Nexin-5 staining. So at 5 and 10 micromolar 6965, A549 cells start rapidly undergoing apoptosis, and this effect is increased dramatically if you treat with the mTOR catalytic inhibitor. Indeed, we head-to-head compared the ability of 3 different mTOR drugs to synergize in their apoptotic potential if we compare them with DMSO or our ULK1 inhibitor or chloroquine, and in every context, combining our ULK1 inhibitor with the mTOR catalytic inhibitors led to the greatest amount of apoptosis. So the take-home messages for ULK1 inhibition then are that for tumors that are reliant on mTOR catalytic activity, So tumors that have mutations either within the mTOR pathway itself or the PI3 kinase pathway. Treatment with rapamycin analogs or other mTOR inhibitors are starting to enter the clinic, and we believe that part of the reason these drugs may be cytostatic is because when you put those inhibitors on, you actually induce autophagy and you induce UK1 activity as a cell survival mechanism. So if you combine rapalogs or other mTOR catalytic inhibitors with UK1 inhibitors, you will improve the therapeutic response. But this need not be limited to mTOR inhibitors. It may be that actually treatment with a wide variety of therapies induce autophagy, and therefore combining a UK1 inhibitor with many different types of conventional and targeted cancer therapies will emerge as a benefit. Finally, though, we also know that there are some subsets of tumors that are truly appear to be addicted to autophagy. And we in the field are learning more and more about how to best define those tumors and the patients that would be benefited by this. But in those cases, UK1 inhibition as a single agent appears to be as effective as many other treatments. So that's another very active area of exploration. So as my final slide, and now some 10 years later of investigation from many labs has really developed a far more complete picture of how AMPK reprograms cell metabolism, and therefore how a wide variety of natural insults to the cell and nutrient deprivation reprograms metabolism. So all these phosphorylation sites shown on this cartoon are all different things that the field is developing reagents to, and different phospho-specific antibodies can help guide you towards whether or not any of these things are important in your favorite tissue or disease state of interest. And as I said, we focused a lot today on UK1. Finally, I would be remiss if I didn't show you a picture of the lucky people in my lab that I am very lucky to have work for me and who have done much of the work that we talked about today. The UK1 project was a inhibitor project was a collaboration with Nick Cosford, shown here in his laboratory at Sanford Burnham, just up the street here. A large number of people from his lab helped out, but some of those are just highlighted here. Within my own lab, the key drivers of the UK1 work were Dan Egan, very talented PhD student in the lab, and Matthew Chun, a postdoc in the lab who helped inherit the project from Dan and then took it on into the therapeutic avenues. At this point, I'd like to thank you for listening to this intense blast of science and that I'm ready to answer questions. Thank you.
Excellent presentation, Dr. Shaw. Thank you for bringing that information to us. Before we get to the questions, I just want to remind our audience that they can submit them through the green Q&A button at the lower left of the presentation window, and we're going to get to as many as we can. Our first question: given that inhibition of autophagy, as in ATG7 knockouts with chloroquine, can inhibit tumor growth, do you predict that ULK1 inhibitors can be used as standalone therapeutic agents, or do you think the cytotoxic effect of these inhibitors relies on the addition of autophagy inducers like mTOR inhibitors?
Okay. Great question. Let's see. Yes, I'm on. Great question. I think I may have touched on this right at the end, but I think, at least from the data that the world has so far, we and other labs have so far, we think there are a subset of tumor types, perhaps pancreatic cancers based on Alec Kimmelman's work, or tumors containing BRAF or KRAS mutations, where ULK1 inhibitors may show the greatest benefit as a single agent. And then for other more complex somatically acquired tumors, such as lung cancers or colon cancers, there you might want to combine it best. That's the way that it will be used. Great.
Thank you for answering that. Our next question: you have shown that AMPK inhibitors AMPK inhibitors I'm sorry, I've lost the question. Our next question, Hee-Jong from University of Nebraska asks, what is upstream of LKB1 and how is LKB1 activated?
Great question. Great question. The whole field has actually been searching for answers to that question for some time. The thought in the field right now is that LKB1, I should say the dogma right this minute, is that LKB1 is just active in a wide variety of conditions normally. So in tissues, and certainly my own lab has seen in the liver, for example, of mice, without insult, LKB1 is active and is phosphorylating a number of its downstream targets outside of AMPK. So LKB1 phosphorylates other targets besides AMPK. And perhaps it's only under conditions of low ATP that AMPK gets brought to LKB1. I think that question really is one of the biggest ones in the LKB1 field that remains to be answered. We don't know. We wonder whether or not there are phosphatases that actually are stimulating the process to begin with, but whether or not there are other kinases upstream of LKB1, that is not known, frankly. There's a number of different candidates, papers have looked at it, but there's no direct answers. Next question.
Great. Our next question. At a given time in a cell that grows in a nutrient-replete conditions, could a significant fraction of AMPK still be active? If so, could this AMPK subset be sequestered in other protein complexes, for example, ULK1?
Yeah, that's another great question. I think that's another area the field is kind of steering towards now, or at least in my own opinion, people in the field are steering towards, is what are the different pools of AMPK within the cell? And are there some pools, just as your question is getting at, that in fact may be active even when nutrients are there? And so that you always have some mixture of AMPK being on and regulating some subset of targets, perhaps around the mitochondria, but then that other populations are kept inactive. And so I would say the field does not know, but I think that is very plausible, and I think that's going to be a very interesting area to watch, and I hope many different labs in many different systems actually try to explore that. And I think it's very possible that ULK1, AMPK, and mTOR itself at certain times are at certain locations within the cell. And maybe that'll come back in other questions. Next question. Next question.
Great, thank you. Would you consider activation of autophagy in early AMPK activity? Many studies don't see activation of AMPK until 24 to 48 hours after metformin treatment. Do you suggest that there are both early and late stages of AMPK?
Yeah, that's another really good question. I do think, and there is precedent for published data for different waves of AMPK activation. In some studies, there was a very nice MCB paper from maybe 2007. Specifically, you could find it if you looked up leptin, do a PubMed search for leptin in C2C12 cells. And in that study, they were looking at a biphasic AMPK response in response to leptin in that cell type, but they also explored activation of different pools, in fact, of AMPK. So that comes back to one of the earlier questions. And again, I think this is really underexplored. I should finally add, relating to your question though, I think another limitation within the field is that the tools to study AMPK, particularly in tissues, both from animals, certainly in rare tissue settings from human patients, that AMPK actually gets activated when you harvest tissues. So frankly, within seconds, even certainly within minutes of isolating a tissue. You get very robust AMPK activation in the tissues before you flash freeze them. So that has confounded many different studies looking at AMPK activation after given drugs. In our own hands, we can see AMPK getting activated in rodent liver within 30 minutes of metformin treatment. So I think a lot of it is really being able to get the background that is from harvesting reduced to see those effects. So we think metformin very rapidly induces AMPK in the body, depending on the tissue you're looking at. And in hepatocytes, for example, it activates AMPK within 15 minutes in primary hepatocytes. But your concept of different waves of AMPK, I think, even though there's not great data for it, I'm sure that's true. Why don't maybe we can try to get some questions from try to get questions from different people since we're running short on time. Let's see.
Thank you. Our next question, Josh asks, there is talk of AMPK being a context-dependent oncogene. Is it promoting autophagy in established tumors? If so, why not develop AMPK inhibitors?
That's another great question. And I think there is emerging data. There was actually a very nice paper, which I believe is just online in the past couple of weeks. In fact, you'll see there's an accompanying news and views from that I wrote for this paper, in cell stem cell, genetically proving within a subtype of leukemias that AMPK is in fact required for the growth of those leukemias. So this is one of the first well-defined examples of a context-dependent oncogenic activity. What's even more remarkable about that study is that AMPK is only having the function in certain tissue sites. So AMPK is only active in those cells when they're in the bone marrow, not in the spleen or in the blood in other settings. So it illustrates some of the complexities that are going to be involved in when AMPK or ULK1 itself may be engaged physiologically to keep tumor cells alive. Again, your concept, though, is valid that you might want to develop AMPK inhibitors. I assume people are doing so. And I think that the challenge there, just like ULK1 inhibitors, you'll only want to use them in settings that are short. You don't want to incidentally give someone a different disease just by treating their cancer. But I think there is the prospect. So good question.
Thank you for that answer. Our next question from Sumeta, can AMPK activate autophagy in the presence of active mTORC1?
Good question. That has been explored mostly in cells, of course, in the context of treating with different doses of AMPK activators, which themselves will lead to a decrease in mTOR. I think a different way to rephrase your question, within the cell at any given time, when you treat with either a pharmacological drug to activate AMPK or under nutrient deprivation conditions, you will have individual AMPK complexes floating around the cell that are going around and shutting off mTOR and going around and activating ULK1. So I think there is a battle at different sites within individual cells that are going on. And the net output of how much autophagy is induced, I think that is an open question. However, there are examples, at least just from studying published reports, where you have cell conditions where AMPK appears to be active, as read out by some of its own phosphorylation or phosphorylation of its targets. mTOR is also active in the same lysate, at least, but the dominant effect is a suppression of autophagy. So either the relative activation, TOR, is just much higher. Even though you see some AMPK, TOR is winning the battle within individual cells. Or you have within different populations, within the tissue or within different cells, AMPK has won the battle and it's winning it. It is inhibiting autophagy within a subset of the cells, whereas in another subset, TOR is high. This is going to be something that's going to take single cell analysis to really answer. But I think with better tools to look at autophagy, that we'll get there.
Great. Our next question from Naveen: which tumors or cell lines are the most dependent on autophagy?
Yeah, that's a good question. For that, I would actually refer people to published works, probably the work of Alec Kimmelman. There was a genes in development paper from his lab, so Kimmelman, that looked at this direct question, at least using chloroquine as a readout of autophagy inhibition, and it's one of the first figures of his paper. But there are other papers that have looked at this. My own lab has not really looked at this that much in depth, nor have we fully, some of the other questions later on that we're not going to have time for, we have not fully examined ourselves massive cell line panels to actually ask where our compound may work best as a single agent. So those are ongoing things. The true settings where 6965 will be useful in different subsets of cancer and in mice and all those kinds of things, those are all ongoing types of studies. So I think in the interest of time, let's just take, I think we really only have time for one or two more questions. If we can go to question 18 here, I guess it's from the same individual, but that's another interesting question.
Great. Thank you. What type of nutrient deprivation do you perform to induce autophagy in your systems? Zero glucose or low glucose, or zero serum or low serum, glutamine limitation, glutamine absence, or some combination? So for this, I would refer people to that recent publication, the molecular cell paper from my lab, Egan, Chun et al. In most of the settings, we used EBSS media, which is basically amino acid deficient media, but it still has glucose. But we can also get the same effects by putting cells into no glucose. It just takes a longer amount of time. So the fastest way to induce ULK1 or to induce autophagy in many people's hands is either with EBSS media or HBSS media. In my own lab, we use EBSS. So let's go to potentially a final question, question number 2. Great, thank you. Have you looked at 6965 effects in cancer models in vivo, pancreatic cancer in particular? Okay, that's actually question 22, but that's fine. I meant question 2, but we'll just end here on this note. As I alluded to in my answer to the previous question, that is something that we are actively looking at right now. So if any of you see me at a meeting or someplace else, ask me that question again in 6 months, and we'll see where we're at. But that's something that multiple people in the lab are actively pursuing. Thank you. Great. Thank you so much. I'm just trying to find your last question too. You have shown that AMPK inhibits mTOR and that many targets such as LKB1 are regulated by the two kinases in a yin-yang manner. Can mTOR directly inhibit AMPK and how?
Yep. Okay. So I think this is another interesting area for the field. The question is, can mTOR directly, how does mTOR itself inhibit AMPK? If AMPK is going around the cell trying to shut off mTOR, is mTOR itself trying to directly inhibit AMPK? So there are no direct phosphorylation sites from the TOR complex itself known within AMPK. However, there are reported S6 kinase sites within AMPK. In fact, some of the same sites that in other conditions are thought to be phosphorylated by AKT to inhibit AMPK function. And on that topic, I would refer you to a study from Barbara Kahn's lab in the journal Cell Metabolism, I think about 2 years ago now, reporting that S6 kinase 1, a direct mTOR target, can directly phosphorylate one of the two catalytic subunits. So with that, thank you for all this great interest and feedback. This is the first time I've ever done a webinar, and I can see this is a very exciting and fantastic way to connect researchers with the broader research audience. So I hope all of you enjoyed it, and I think there'll be a mechanism for further feedback as well. But I hope all of you, wherever you are, all over the world, have a great rest of your day. Have a great night. Thank you.
Thank you, Dr. Shaw, for bringing that information to us. Excellent presentation today. I do want to let our audience know that this presentation will be available on demand through May of 2016. They're welcome to log in and look at it again. You'll be receiving an email from LabRoots when it's available, and we invite you to share that email with your colleagues. I'd like to also thank our sponsor, Cell Signaling Technology, for making it possible to bring this information to you today. Thank you again for joining us. Goodbye.